This video focuses on meiosis, which is that form of cell division that's used in sexually reproducing organisms to produce their reproductive cells. But before we get into the details of how meiosis occurs, it'll be useful to spend some time thinking about and comparing asexual reproduction and sexual reproduction more broadly. We should differentiate between the terms sex and reproduction, because even though we tend to think about these things as always being inextricably linked, because that's the way it occurs in the animal world, we should recognize that these two terms are talking about different things. Reproduction specifically refers to the creation of new individuals in the population, new organisms, whereas sex is really referring to the exchange or combination of genetic information. They are two distinct things. In animals like us, these two things are linked because we do reproduce sexually rather than asexually, but it's not always the case. In fact, even bacterial cells that reproduce via the asexual process of binary fission still find ways of exchanging genetic information with one another through a process called conjugation. In this process, two bacterial cells, each of which has its own DNA, consisting of its main chromosome as well as extra chromosomal plasmids, would generate what's called a sex pilus, which is essentially a little protein bridge that connects the cytoplasm of these two cells, and in so doing they would be able to pass DNA molecules, generally these plasmids, back and forth and exchange genetic information. In that way they generate genetic diversity, which is so important for increasing fitness in the environment. If we're talking about multicellular animals and plants, these organisms would have specialized reproductive organs that would produce gametes or reproductive cells, which would then combine and produce new individuals in the population. All organisms, no matter what domain of life we're talking about, are going to have means of generating genetic diversity and exchanging DNA with other organisms in the population, but that's not necessarily linked to their mode of reproduction. We've talked before about binary fission and mitosis as means for single-celled organisms to reproduce themselves via an asexual mechanism, generating identical clones from one parent cell. But we should note that there are lots of ways for organisms to reproduce in an asexual fashion besides binary fission and mitosis. Here we see an example of budding in a hydra, which is an aquatic organism. What you see here is the parent hydra budding off a little juvenile hydra, which, when it's mature enough, will actually break off from the parent and then go on as its own independent free-living organism. That's asexual reproduction because this bud is actually a clone of the parent. If you garden, you've seen plants like strawberries send out runners and new plants emerging from particular nodes on those runners. That's a form of asexual reproduction. All of these new plants are going to be clones of the parent. Anytime you grow a plant from a tuber like a potato, these sprouts that generate the new plant are clones of the original parent plant that produced that tuber. So that would also be an example of asexual reproduction. One of the most extreme examples of this mode of reproduction is the colony of quaking aspens in Utah referred to as Pando. This is generally considered to be the largest organism on Earth because what this stand of trees actually is one colony that is all connected by a common underground root system. And the way that all these independent trees have sprung up is by suckers that originated from the original parent tree. So technically we can consider them all part of one organism because they share an underground root system. But if something happened to sever one of these shoots from surrounding trees, it would be able to live as a free-living organism, so it's actually another organism in the colony. Take-home message: there are lots of ways that organisms can reproduce in an asexual fashion, and there are definite advantages and disadvantages associated with this mode of reproduction. For example, if you're a non-motile organism like a plant that can't move, finding a mating partner is not easy. Plants accomplish sexual reproduction by producing an excess of gametes that they can then distribute to the environment. So the gametes themselves find the mating partner. An example would be trees or other plants producing pollen that gets distributed on the wind or via other pollinators. It takes a lot of energy to make all of those gametes. So if you are able to reproduce via an asexual mechanism, then you don't have to waste that energy making an excess of gametes to distribute to the environment. The other thing is that if you reproduce via a mechanism like sending out runners or shoots or suckers, then the offspring are actually going to be multicellular and a bit less fragile than embryos that are sprouting from seeds. This results in offspring that are a bit more sturdy, that are going to be able to withstand the environmental stresses of growing in their new location. Another advantage is that if the parent is well adapted to the environment, then because the offspring are clones, they are going to be equally well adapted to that environment. And that allows a rapid colonization to outcompete any individuals of other species or offspring of other individuals in the population within that same geographical region. So in terms of competition, you have an advantage. But there are disadvantages associated with the asexual reproduction as well, and many of them are sort of the converse of these ideas that we just talked about. If the parent is well adapted to the environment, then the offspring are equally well adapted. But what if the environment changes and the parent is no longer well adapted to that environment? As you can imagine, that's going to put the entire population descended from that parent organism at a disadvantage in that specific environment. What about sexual reproduction? What we see here is that there's a genetic contribution from more than one parent, and the offspring in sexual reproduction are genetically distinct from one another and diverse within the population. Humans offer a nice illustration of that kind of diversity. We have a lot of diversity in superficial traits and characteristics, so it's really obvious to see that kind of diversity that arises from sexual reproduction. But we should recognize that in addition to these superficial physical characteristics that don't have a whole lot of physiological significance, all of our hidden traits are subject to the same level of diversity. Things like energy utilization and efficiency of metabolism, fertility, susceptibility to diseases like cancer or diabetes. All of that is subject to the same level of diversity. We just can't see it on the surface. Though I showed you several examples of plants reproducing via asexual reproduction, most plants primarily use sexual reproduction as a means of generating new individuals in the population. They have specialized reproductive organs just like animals do. Those would be flowers if we're talking about angiosperms, or pine cones if we're talking about conifers or gymnosperms. These specialized organs produce specialized reproductive cells or gametes that can combine and produce new individuals in the population. That contribution from more than one parent generates a lot of genetic diversity, as we've discussed. In order for sexual reproduction to take place, there has to be that specialized population of reproductive cells, the gametes. If we're talking about animals, sperm and egg, or ova. If we're talking about plants, pollen and ova. The form of cell division that's used to generate those gametes is, of course, meiosis. Before we talk about the details of meiosis, we need to spend some time talking about DNA and the change in DNA content that's necessitated by using sexual reproduction as a mode of generating new individuals in the population. Take an example of a cell here that is from some organism that reproduces via sexual reproduction. This is a diploid organism just like us. Ploid or ploidy refers to chromosome number. Di, as we've seen before, means 2. So this would be an organism that has 2 sets of its chromosomes. We are also diploid organisms. This is just a simplified model of this situation we would see in ourselves. The term haploid refers to half. So a haploid cell is one that has half the DNA compared to the cell that produced it. Meiosis is a reductive division. We start with a diploid cell, and that diploid cell divides and produces reproductive cells that have half of the DNA content of that original starting parent cell. With half of that DNA content, it can now combine with another haploid cell to produce the new zygote or offspring from that reproductive event. In this example, we're looking at a cell that has 2 sets of 3 types of chromosomes or DNA molecules. We can distinguish them as large, medium, and small. If this is chromosome large, there's another chromosome large that would have been inherited from the other parent. This is indicated by the color coding here, where red refers to all of the chromosomes that were inherited from the maternal parent of this organism, and blue is indicating all of the chromosomes that were inherited from the paternal parent of this organism. In other words, mom and dad. By convention, the chromosomes are numbered from largest to smallest. This chromosome 1 and this chromosome 1 are referred to as the same type of chromosome because they have the same sets of genes on them. So that's chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from mom, chromosome 2 from dad, chromosome 3 from mom, chromosome 3 from dad. We call those chromosomes of the same type, one inherited from each parent, homologues or homologous chromosomes. In this case, we're looking at a cell that's preparing to undergo cell division. It has already been through S phase, And we know that the cell has already been through S phase because what we see here are sister chromatids that are joined together at the centromere by cohesin proteins. So in this case, we're looking at a diploid cell that contains 6 chromosomes total. The diploid chromosome number, 2n, meaning 2 sets of n types of chromosomes, is equal to 6. The haploid chromosome number n, that would be in 1 complete set of DNA, is 3 for this cell. For comparison, if we were talking about our cells, we have 23 types of chromosomes. Since we're diploid organisms, we inherit 1 set from mom and 1 set from dad. So we have 2 sets of 23. Our haploid number, n, is 23. Our diploid number, 2n, is 46. So if we were looking at a human cell here, we wouldn't see 3 pairs of chromosomes, but 23 pairs of chromosomes. So this is just a simplified scenario to help us visualize what's going on. These homologues have the same genes, but that doesn't mean that the DNA sequence all along these chromosomes is identical. We know that genetic mutations occur constantly and generate new mutations in the population. So at many positions along this DNA sequence, which is hundreds of millions of nucleotide base pairs long, we're going to see differences in the sequence between this chromosome 1 inherited from mom and this chromosome 1 inherited from dad. But the genes that are on these chromosomes are the same. In other words, if at this position on chromosome 1, there is a gene that is associated with hair color, at that same position on chromosome 1 inherited from mom, there's also going to be a gene for hair color. Does that mean that the hair color gene sequence is going to be identical on both of these sets of DNA? No, the gene that's inherited from dad might encode red hair. Maybe the gene that's inherited from mom might encode black hair. So same gene, but differences in DNA sequence cause those genes to encode different proteins, and those proteins are associated with different traits or characteristics for whatever this gene specifies. These alternate versions of the same gene that differ in sequence are referred to as alleles. So homologues are the chromosomes of the same type with the same sets of genes, but potentially different alleles of those genes. These terms, chromatid, homologue, and allele, can be very confusing, so you need to spend some time making sure that you understand the distinctions between them. Just to reiterate, sister chromatids are the identical copies of DNA produced during S phase, during DNA replication. Homologues are the chromosomes of the same type that have the same sets of genes on them, one inherited from each parent. And alleles are slightly different versions of the same gene that arise from random mutations in the DNA. Human cells have 46 chromosomes, and we can visualize those chromosomes using what's referred to as a karyotype. A karyotype is generally created using what's called a metaphase squash, in which cells that have been artificially stalled or paused in metaphase when the chromosomes are in their most condensed state are then exposed to a dye that binds to the chromosomes. Then a picture is taken to display that full set of chromosomes. In humans, the way that you would do this kind of metaphase squash is you would take a blood sample and treat the blood sample with a compound that induces white blood cells to undergo mitosis. Then you treat them with another drug that actually causes mitosis to stall right in the middle of metaphase. This drug disrupts the microtubule network so the cells can't get past metaphase. Then you literally squash the cells to spread the chromosomes out from one another and take a picture. Once upon a time, this would have been a picture on real film, and you'd cut out all the individual chromosomes and line them up manually. Now, of course, this is all done digitally. The dyes that are used in this procedure will bind to different chromosomes differently to produce unique banding patterns. Here we're looking at a karyotype of all of the chromosomes in a full set of the human genome. We have 23 pairs of chromosomes. 22 of them are what are referred to as autosomes. These are all of the chromosomes other than the sex chromosomes. Then we have the one pair of sex chromosomes, the X and the Y. The dyes that are used for karyotyping will bind differently to each of these different chromosomes based on differences in chromatin structure, and experienced karyotypers can look at this and very quickly identify which chromosome they're looking at, just by the banding patterns that are observed on those chromosomes. This can detect gross genetic abnormalities, things like changes in chromosome number. So instead of being diploid, maybe you inherit an extra copy of one of these chromosomes and you're actually triploid for that chromosome set. Or you're missing a chromosome for one of these. There's a chromosomal deletion. It can also detect deletions and insertions within a chromosome. Let's say there's a chunk of this chromosome here that's missing. Or a hunk of one chromosome gets moved from one chromosome to another, which would be called a translocation. This would not reveal small-scale changes like differences in single nucleotide pairs within the DNA sequence, but it's a really useful technique for visualizing large-scale abnormalities in chromosome structure or chromosome number. So how do we end up inheriting exactly one set of DNA from each of our parents to end up with this karyotype? Like all animals, we have reproductive organs that contain specialized cell populations that generate gametes or reproductive cells with half the genetic content of every other cell in our body, and that occurs through meiosis. This is the mechanism for producing sperm and egg, which then combine through the process of fertilization to produce a new zygote, the earliest stage of embryogenesis, which is diploid once again. Then the way we get from that very early embryo to the adult stage is massive waves of mitotic cell division that drive embryonic growth and development and specialization of those resulting cells to make all of the tens of trillions of cells in the body. This is how we control chromosome number across the generations. If you just took a random cell in the body that reproduces via mitosis and combined it with another cell from another individual in the population, then obviously you would be doubling your chromosome number every time two individuals reproduced. But in this way, we cut our chromosome number in half. Then, when those cells combine with one another, we get back to our original chromosome number once again. Let's compare that change in chromosome content with what we would see happening in mitosis in one of the cells of our bodies. In mitosis, a cell is dividing to produce identical daughter cells. We're not talking here about reproduction in the sense of producing new organisms, just reproduction at the level of the cell. This could be happening in a liver cell, or in a skin cell, or a bone cell. Mitosis is going to produce new daughter cells. We would start with a diploid cell with 46 chromosomes. Before that cell goes through mitosis, it's going to go through S phase. Each chromosome gets duplicated, so that's going to give us 92 DNA molecules. Then the cell would go through mitosis, and what we would end up with is two daughter cells, each of which have 46 chromosomes. So we get right back to where we started. And these daughter cells are identical clones of the parent cell that we began with. Now let's contrast that with what happens with meiosis. Meiosis is only going to happen in that specialized population of cells that is present within the gonad, so in ovary or testes. That germ cell is diploid like all the other cells in the body. It's going to have 46 DNA molecules. Those 46 DNA molecules are all going to be copied in S phase. But in contrast to what we see happening up here with mitosis, with meiosis we see one replication of DNA, but then two rounds of cell division. So we have meiosis 1, abbreviating that as M1, which is going to cut the chromosome number in half. And then each of those cells will undergo a second cell division event, meiosis 2, or M2, resulting in half the DNA content that we began with. One DNA replication, two cell divisions, and we end up with 4 haploid daughter cells compared to the original parent. So chromosome number has been halved, but the other thing that we're going to be seeing is that during meiosis, more specifically during meiosis 1, we are going to see the introduction of genetic variation that's going to make each of these daughter cells genetically distinct from one another and from the cell that we began with. First, let's look at an overall summary of what happens during meiosis. Let's say we have a cell with these two homologous chromosomes. In interphase, the cell will go through S phase. Each of these is going to be duplicated, so we would now call these duplicated homologues. Each has been copied and now consists of two sister chromatids held together by cohesin proteins. In the first meiotic division, these two duplicated homologues are paired up and then sent to two different daughter cells. They get separated from one another. Then, in meiosis II, the cohesin proteins that are holding these sister chromatids together are broken and the sister chromatids are separated. In meiosis I, duplicated homologues are separated. In meiosis II, sister chromatids are separated, and the haploid cells that we end up with have half the genetic content of the diploid cell that we started with. Now that we've seen an overview of what happens during meiosis, let's delve into the details a little bit more. We're going to focus first on meiosis I. Mechanistically, what we see happening in meiosis and mitosis is kind of similar in terms of the condensation of chromosomes, fragmentation of the nuclear envelope, chromosome separation that's being controlled by microtubules, which themselves are being organized by the centrosome and microtubule organizing center. But an important difference is that during meiosis I, genetic diversity gets introduced into the gamete. And another is that overall we see a reduction in chromosome number through these two cell divisions. Let's take a look at meiosis I in more detail, and we'll be focusing in particular on what is different between meiosis and mitosis. As the cell goes from interphase and enters prophase of meiosis I, what we call prophase I, what's happening is very different from what we see happening during mitotic prophase. In meiosis, the duplicated homologues, chromosome 2 and chromosome 2, chromosome 1 and chromosome 1, chromosome 3 and chromosome 3, all get paired up to form what are called tetrads. These are called tetrads because we've got two duplicated chromosomes, each of which is comprised of two individual chromatids, which adds up to a total of 4 DNA molecules that are being held together. Once these homologous pairs have been brought together in the cell, the non-sister chromatid arms exchange bits of DNA, and the points at which this exchange is going to happen are called chiasmata, or chiasma in the plural. What actually happens here is that these DNA molecules are matched up due to sequence similarity and induced to break at this position and then rejoined or ligated such that this little bit of blue that was originally part of this chromosome is now combined with this red chromosome, and this little bit of red that was once part of this chromosome is now joined to the blue. This is referred to as crossing over or recombination. Chiasmata, or crossover points, are the points where the DNA gets broken and then rejoined, but the DNA has swapped places. What's illustrated here is a very simplistic scenario where we have 2 points of crossover on this chromosome. But if we look at actual chromosomes that are undergoing prophase I, what we would see is numerous crossover points all along the length of this paired set of homologues. And in contrast to what we see here, it wouldn't just be these two middle non-sister chromatids that are exchanging DNA, but these outer ones as well. What this does for the cell is generate completely unique sets of chromosomes with new combinations of alleles. Imagine that on this homologous pair of chromosomes, we've got hair color alleles up at this end of the chromosome. This allele coding for red hair, this allele coding for black hair. And let's say that we've got a gene for eye color down here. And on this chromosome, green eyes are what are encoded. And down here at this position on the homologue, we've got brown eyes encoded. On this parental chromosome, we have a combination of black hair with brown eyes. And on this parental chromosome, we have a combination of red hair and green eyes. If there's a crossover event such that these two chromosomes exchange this leg of DNA, then what we would now have is red hair combined with brown eyes, and black hair combined with green eyes, entirely new combinations of alleles that weren't present on the original chromosome. So we get that genetic diversity among the gametes. After this, the molecular events are similar to what happens in mitosis. We see the spindle forming, the nuclear envelope fragmenting, and the microtubule motor proteins linking up to the kinetochore proteins on the duplicated homologues. But the way that these microtubules join up to these chromosomes is a little bit different than what we saw in mitosis. Remember that in mitosis, the microtubules from opposite poles get connected to each of the sister chromatids. But in meiosis I, those are going to get connected to each duplicated homologue in the tetrad. There will be kinetochores on both of the sister chromatids, just like we had before, but the microtubules join up in such a way that microtubules from one pole are going to be grabbing onto this whole duplicated homologue, and microtubules from the other pole are going to be grabbing onto this whole duplicated homologue. In mitosis, all of the chromosomes lined up single file, with the two sister chromatids being connected up to the opposite poles of the cell. But in meiosis, the whole tetrad gets lined up single file, and the tug of war is going to pull that tetrad to the midline of the cell. There's a separate set of proteins that's referred to as the synaptonemal complex that joins these duplicated homologues together in prophase of meiosis I and keeps them together through metaphase until they separate in anaphase. And those are different from cohesins. It's a completely separate molecular complex that holds these two duplicated homologues together to form this tetrad. Once again, the tetrads are all aligned, with these motor proteins trying to pull that duplicated homologue in that direction, and motor proteins over here trying to pull this duplicated homologue in this direction. But as long as this complex of proteins between them is not allowed to be cleaved, then they're not going to make any headway. Just like we saw in mitosis, we're going to see alignment at the midline of the cell due to that tug of war. When we see that, we say that the cell is in metaphase I of meiosis, or is in metaphase of meiosis I. Notice here that our paternal and maternal chromosomes each line up independently of every other homologue set. So if this is the homologue set of chromosome 1, and this is 2 and this is 3, notice that for chromosome 1, the originally paternal chromosome in blue is oriented such that it's going to be pulled to the top, and chromosomes 2 and 3 that were originally derived from the paternal parent are oriented such that they're going to be pulled to the bottom, and vice versa for these red chromosomes that originated from the maternal side. This random alignment and independent assortment of these homologue pairs from one another is another point that generates genetic diversity, because each of these homologue pairs could align in one of two orientations. So to recap, we see genetic diversity generated two ways during meiosis I. The first way is through recombination or crossing over during prophase I, and the second way is through random alignment of each of these tetrad sets at the metaphase plate during metaphase I. Just like we saw in mitosis, there's a checkpoint that prevents the cell from going any further until all homologue pairs are lined up and equal tension is sensed. Once equal tension is established, this synaptonemal complex is cleaved, and the duplicated homologues are separated from one another and pulled to opposite poles of the cell. What we end up with is two daughter cells. One daughter cell contains this set of homologues, and the other daughter cell is going to contain that set of homologues. And then we'll see a cytokinesis event that's going to result in two new daughter cells. Notice that the sister chromatids have not been allowed to separate at this division. They're still being held together by those cohesin proteins. What happens next is the second meiotic division, during which these sister chromatids do get separated. Meiosis II happens exactly like mitosis does. If the nuclear envelope has completely reformed and the spindle has gone away, which doesn't always happen, it depends on the cell type, we're going to see the onset of prophase once again, where the chromosomes condense, the nuclear envelope fragments, we see the spindle forming, we start to see microtubules forming attachments to the kinetochores on our sister chromatids. The motor proteins pull in opposite directions, and these chromosomes are all going to be aligned single file at the equator of the cell. Once equal tension has been sensed on those kinetochores, the cohesins are cleaved, and the motor proteins separate these chromatids. Then telophase and cytokinesis occur, resulting in two daughter cells, each of which has one set of 3 chromosomes. Notice in the diagram, the DNA in these cells are combinations of blue and red. So what we end up with are 4 haploid daughter cells, all of which are genetically distinct from one another, with DNA molecules that are actually distinct from the DNA that we started with because of recombination. Zooming back out and comparing in broad terms what's happening in mitosis to what happens in meiosis, in both cases we start with a parent cell that has 2 sets of 3 types of chromosomes, a diploid cell with 2n equal to 6. Before the onset of cell division, in both cases, the cell has to copy its DNA. So this cell on the left is a somatic or non-reproductive cell. It goes through S phase. All of these chromosomes are duplicated. Then, in prophase, prometaphase, the nuclear envelope breaks down, the chromosomes condense, microtubules form their attachments to the kinetochores, Motor proteins pull on and align those chromosomes. At this point, notice that each duplicated homologue independently aligns single file at the cell equator. When equal tension is sensed, the cohesins are cleaved, and all of these sister chromatids are pulled in opposite directions. The cells we end up with are diploid, just like the parent cell that we started with, and are identical in every way. On the other hand, if this was a germ cell in the same organism, then the events are going to be different. The cell starts with having to duplicate the DNA, just like for mitosis, but following that we're going to have two meiotic divisions. In the first meiotic division, in prophase, the matching pairs of homologues are brought together to form tetrads. We see those chiasma, those sites of crossing over, recombination takes place, and metaphase sees the alignment of tetrads, randomly with respect to one another at the metaphase plate. What gets separated in meiosis I are homologues. The second round of cell division is where we see cohesin being cleaved and sister chromatids separating. The result is 4 daughter cells that each have only one set of genetic information, not 2. Notice that they are all distinct from one another with respect to the DNA molecules that they contain because of those recombination and random assortment events that happened in prophase and metaphase of meiosis I. So mitosis gives us 2 genetically identical diploid daughter cells, and meiosis gives us 4 genetically different haploid daughter cells. Don't forget the context where this is happening. If this was a female, meiosis would have been happening within the ovary, and these cells that are produced would be eggs or ova. These are now the gametes that could potentially be fertilized through union with a spermatozoa to form a new embryo, a new organism. Now that we've talked about the details of meiosis, and we've seen how the gametes that are used in sexual reproduction are produced, I want to return briefly to a more broad discussion of sexual reproduction and asexual reproduction and their evolution for just a bit. Just as we talked about with asexual reproduction, there are advantages and disadvantages associated with sexual reproduction too. What disadvantages can you think of? Well, they're sort of the corollary of the advantages of asexual reproduction. If you are not a motile organism, you can't really move around and find a mate. And so what you have to do is distribute a huge excess of gametes to the environment in some way, and hope that your gametes find an appropriate partner cell to combine with. The other thing that we saw that was an advantage of asexual reproduction is that if you are particularly well adapted to your environment, then your offspring are equally well adapted. Well, that's not going to be the case with sexual reproduction because the offspring are going to be genetically distinct from the parent organism. So just because you're particularly well adapted to your environment, if you reproduce via sexual reproduction, your offspring may not be. So that's a potential disadvantage. Sexual reproduction is slower. It's harder to outcompete other organisms, especially if they're reproducing by asexual reproduction. And finally, if we think about it, those tiny little zygotic embryos are quite fragile. So pretty much every advantage that's associated with asexual reproduction is a potential disadvantage of sexual reproduction. But there's also something else to consider, and that's something that's referred to as the twofold cost of sex. If you are reproducing via an asexual mechanism, your offspring inherit 100% of your genes. But if you combine your genetic information with another individual's genetic information, each of you is only going to be able to contribute half of your DNA to that new organism. Each haploid gamete only contains half of the parent's DNA. So that's one cost that's associated with sexual reproduction. The second cost that's associated with sexual reproduction, in particular for those organisms that have distinct males and females, where only the female organisms gestate to produce the offspring, like we see with animals, in that case, the cost that's associated with sexual reproduction is that half of the individuals in your population don't actually produce new organisms. They contribute a set of DNA to those new organisms, but they can't actually gestate and make babies. In light of that, given that there are these disadvantages, and compared to asexual reproduction, a huge cost associated with sex, how did it ever evolve in the first place? It seems like it would have been selected against. The answer comes down to two things. Number one, It's likely that having a backup set of DNA, in other words, 2 or more sets of DNA, was an advantage to early organisms in case of mutation. The evidence seems to indicate that the recombination we see happening in prophase 1 actually evolved as a mechanism to repair DNA. In cases where one set of DNA was damaged or had mutations that couldn't be repaired, the other DNA molecule would actually be recombined with that damaged DNA to repair it. The molecular mechanisms that are involved with that DNA repair and with recombination during meiosis I are virtually identical to one another. So it's very likely that DNA repair mechanism came first, and then recombination in gametogenesis came second. But the other thing is that diversity generated by sexual reproduction, both during meiotic division, those new combinations of alleles, and also through random fertilization. The particular gametes from any 2 parents that are going to be combined is random. So we're constantly combining alleles in new combinations, and just by chance, occasionally we may combine particularly beneficial alleles in one single individual, and that individual is therefore going to have a very strong selective advantage in the population. In evolutionary terms, the other hypothesis that's gaining a lot of ground and a lot of research evidence to support it is that a primary benefit of the allelic diversity that we get through sexual reproduction is that we're able to outcompete our parasites and pathogens in a much more efficient manner. We could be talking about viruses, bacteria, fungi, protozoa, anything that uses us as a host and has the potential to be pathogenic and to cause us disease or otherwise cause us harm. The way those pathogens gain entry into our body cells and tissues is through molecular interactions. Our cells have particular receptors that a virus, for example, might hijack in order to gain entry into the cell and to turn it into a virus factory. We experience mutations in our cells constantly, and some of those mutations might change the structures of those receptors, even very slightly, such that those viruses can no longer bind. At that point, you might think, hooray, we won the war. But what's going to happen is that the virus will also accumulate mutations. And some of those are going to allow them to bind to the new receptors. So we undergo mutation, they undergo mutation. It's this constant arms race between us and our parasites. It's only the constant generation of new genetic diversity and allelic diversity that allows us to win that arms race. This is referred to as the Red Queen hypothesis, and this is a literary reference to a passage in Through the Looking Glass. So Alice in Wonderland is talking to the Red Queen, and no matter what Alice does, no matter which direction she goes, she ends up right back where she started. And the Red Queen explains to her, now here you see, it takes all the running you can do to keep in the same place. And that describes very nicely the status that we have with our own potentially pathogenic parasites. Every time we undergo mutation to give us an advantage, they undergo mutation to give them the advantage, and so on and so forth. So that diversity that we get through new combinations of alleles is really, really important in the context of our interactions with other organisms that are trying to gain access and entry into our body cells. This video has discussed some evolutionary context and advantages and disadvantages associated with asexual and sexual modes of reproduction, and provided a detailed look at the mechanism of meiotic division, as well as highlighted the sources of genetic diversity that arise through it. In the next video, I'll talk about what happens when meiosis doesn't occur as it should, and the evolutionary significance of those mistakes.